In our last video, we talked about what an enzyme is, and we talked about the fact that enzymes are extremely important for making reactions occur because they decrease something called activation energy. Essentially, what they do is they make it easier for the reaction to take place. I gave you the comparison of starting a fire. If you try to start a fire with two sticks, you might eventually get a fire, but it's going to take you forever, and at that point, you probably don't need the fire anymore. Or you can start it with something like gasoline or a fire starter, which makes it happen much quicker, and then you get your fire. That is essentially how an enzyme works in the sense that it makes it easier for a reaction to occur. What we're going to talk about today is how does it actually do that. So let's get started. What are the characteristics of enzymes? Well, there's a couple important characteristics. The first one that is extremely important is that enzymes are specific, meaning they only work in one type of reaction. You can't have an enzyme from a different part of the body work on a part that it's not built for. They call it a lock and key relationship. Think of your key for your house, right? You have a key to your door. I can't take my key and open your house. That would be weird, and luckily it doesn't work that way, right? Every key has a certain lock in which it fits in, and then it opens that lock. That is exactly how enzymes work. Every enzyme has a specific shape, and it fits with a specific reaction, and that's it. It only works in that one place. So that's called a lock and key, and that's very important. Enzymes are only able to work in certain instances. Also, enzymes are reusable and recyclable. Enzymes can be used over and over again. They make the reaction start, and then they go and do another one. So I gave you the example of like your friend that likes to start fights, right? They can hop around and get a whole bunch of fights going. They don't actually get in any of the fights themselves, but they can just keep getting other fights going, and that is exactly how enzymes work. They can be used multiple times, and they can be used for multiple reactions. They keep getting used over and over again. And the third very important thing about enzymes is that they only work in a very, very narrow temperature and pH range. Our body temperature is about 98.6 degrees, and our pH is a little bit over 7. That is not a coincidence. The reason why our body temperature is 98.6 degrees and our pH is around 7 is because that is the pH and temperature at which our enzymes work the best. If our enzymes worked better at 80 degrees, well, guess what? That would be our body temperature. So enzymes only work in this very specific range of temperature and pH, which we're going to talk more about in a little bit. So what are enzymes? They're very specific. They can only work in certain instances. They are reusable, and they are only working within a specific temperature range. How do they actually work? Well, let's talk about that now. Okay, so how do enzymes work? Enzymes work by using the following things. You need the following things. You need an enzyme. Obviously, in order for an enzyme to work, you need an enzyme. You need something called a substrate. And the substrate is what the enzyme acts on. That's the reactants. Those are the things that go in. And then you also need favorable conditions. It needs to be the right pH, and it needs to be the right temperature, and it needs to be the right environment. So let's just talk about how it works. The substrate is the thing that it acts on, right? So that's the actual sugar, let's say. If I use lactose as my example. Lactose sugar is in milk. The enzyme attaches to it, and it forms something called the enzyme substrate complex. That is when it attaches to the actual substrate, and it does its job. And when it does that, it breaks it into something new. So let me just show you what that looks like over here. So let's say I have my enzyme here. This is my enzyme over here. This is my substrate. Let's say it is lactose sugar, right? So lactose sugar is made up of glucose and galactose. Our body can't digest it in the form of lactose. We need to break it down. So what happens is the enzyme comes along, it fits right with the substrate, so it attaches to the substrate. What that does is it changes its shape slightly, and it breaks it into two pieces. So what would end up happening is this substrate would end up becoming two separate parts, right? So let's say that trades off. The enzyme does its job. This would be the enzyme substrate complex. And when that enzyme leaves, what you end up with is a molecule that has been broken into two. So now these sugars our body can digest. What does this enzyme do? It goes right back again, and it finds another substrate. And it keeps doing its job. So this enzyme can keep getting used over and over again because it has the ability to be recyclable. So let's just go over that real quick. So what are the steps? So essentially what ends up happening is the substrate bonds to what is called the active site of the enzyme. So what is the active site? That is the place on the enzyme where the substrate attaches. So if I go back to my little diagram here, what is the active site? the active site is going to be right here. That is the active site, because that is where this enzyme is going to be able to attach. So if we're going to my lock and key scenario, it would be the key. It's the place where it would be the lock. It would be the place where the key goes into. So the substrate bonds to the active site of an enzyme, forming something called the enzyme substrate complex. And that's just a fancy way of saying that the enzyme has attached to the substrate. And where it does that it's called its active site. So that's just the location where the enzyme and the substrate attach. So it's the part on the enzyme where the substrate can attach to. When they bind together, it actually changes its shape. Imagine if somebody sat too close to you that you didn't know. Imagine yourself on a park bench. You're sitting there minding your own business, and then some weird stranger comes up and sits right next to you. What are you probably going to do? You're going to move over a little bit on your bench, right? You want to get away from that person a little bit. Well, the same thing happens in molecules. What ends up happening is these molecules attach to each other, and they change their shape slightly. And when they change their shape, a lot of times it breaks those molecules apart. It can also form them together. You can have enzymes that build things. They either build them or they break them down. There's a name for that. It's called the induced fit model. What that means is it changes its shape to fit the molecule. So you get a new structure. And then it changes its shape, it does its job, and then that enzyme goes on to perform another reaction. So it does this process over and over again. So this diagram does a good job of showing that. So let's say molecule A is some substrate. It doesn't matter what it is. It could be lactose, the sugar. It could be a carbohydrate. It could be anything, right? What ends up happening is the enzyme comes along. The active site is the place on the enzyme where that substrate attaches. They form what's called the enzyme substrate complex. When that occurs, there's a change of shape. It changes the molecule into something new. You get your product, and then this enzyme is going to go and start and do a whole nother reaction. So the enzyme just keeps going. It's a cycle. It continues to go on, and that process is going to keep going and keep going. I'm going to show a short animation that explains this.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Okay, so that is it for this